वेलकम बैक टू आवर चैनल बी क्यूरियस। आज हम पढ़ने जा रहे हैं सेल साइकिल एंड सेल डिवीज़न विच इज़ इंक्लूड सेल साइकिल, एम फेज, सिग्निफिकेंस ऑफ माइटोसिस, मियोसिस, सिग्निफिकेंस ऑफ मियोसिस। A small cell give rise to large organism which start life form in single cell. The sequence of event by which a cell duplicates its genome, synthesis the other constituent of the cell and eventually divides into two daughter cells is termed cell cycle. Cell is consist of cytoplasm, nucleus, ribosome, mitochondria, cell membrane. Cell membrane is outer membrane of the cell which give rise to cell division and form two daughter cell. Intact genome is basically the complete, unbroken and functional genetic sequence of an organism or viruses. Scientific view of intactness: full sequence from one that is fragmented or defective, missing critical parts. Biological point of view regarding how DNA persists over time. Phases of cell cycle. The typical eukaryotic cell cycle is illustrated by human cell in culture. This diagram showing that cell cycle. Cell cycle having 4 phases. First is M phase, G1, S, G2. Cell cycle is divided into two basic phases, interphase and M phase. Interphase further divided into G1, S, G2. The M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phase. It is significant to note that in the 24-hour average duration of cell cycle of a human cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of the cell cycle. That means the most part is the interphase, then M phase. The M phase start with the nucleus division. Nuclear division is the starting point of the M phase, corresponding to the separation of the daughter chromosome, which is also called karyokinesis. Place of chromosome represent karyokinesis and usually ends with division of cytoplasm, cytokinesis. And the place of cell division is cytoplasm, called cytokinesis. The interphase thought called the resting phase. Interphase is also called as resting phase. Is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication. Division in M phase, cell growth and DNA replication is occur. The interphase is divided into 3 further phases: G1 phase, which is also called gap 1, S phase, synthesis phase, G2 phase, gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. G1 is the place for mitosis and DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. In G1 phase, its place for growth and not replication its DNA. S or synthesis phase marks the period during which DNA synthesis or replication take place. S phase is the place for synthesis where DNA synthesis occur and the replication take place. During this time, the amount of DNA per cell doubles. Here we notice that DNA is doubled, okay, If the initial amount of DNA is denoted as 2C, then it's increased to 4C. Means it's going to be double. However, there is no increase in the chromosome number. This represents that no increase in the chromosome number, but it can be doubled. If the cell had diploid or 2n number of chromosome at G1, even after S phase the number of chromosome remain the same, 2n. In animal cells, during the S phase, DNA replication begin in the nucleus. DNA replication begin in nucleus and the centriole duplicate in cytoplasm. Okay, there are two places, nucleus and cytoplasm. In nucleus DNA replication occur and in cytoplasm centriole duplication is occur. During the G2 phase, the protein are synthesized in preparation for mitosis while cell growth continue. Okay, phase of cell cycle I discussed further, interphase and M phase. Okay, dividing once in approx 24 hours. For example, yeast progress through the cell cycle in only about 90 minutes. Cell in M phase, cell is divided in nucleus division. Okay, first is karyokinesis and cytokinesis. Separation of daughter chromosome is karyokinesis and division of cytoplasm cytokinesis. इंटरफेज रेस्टिंग फेज भी कहा जाता है, जो कि G1 फेज, S फेज और G2 फेज, गैप 1 सिंथेसिस G2 फेज. और G1 phase is recognized for growth and quality control, and S phase is synthesis, where 2n to 4n DNA going to be term of chromatides, production of protein and organisms. M phase is four types of, further divided into four types: prophase, metaphase, anaphase, telophase. In prophase, it is a preparing stage where chromosome condense, give rise to blueprint tightly packed. In metaphase, it is a middle phase, chromosome aligned at the equator, and then check phase to ensure equal distribution. And the anaphase is away phase, where sister chromatids are pulled apart. Telophase is two or twins phase, where two new nuclei form. Chromosome, CHR is chromosome. Phase G1 and S, G2, M are the phase of the M phase, which is primary activity. G1 is growth and key event is a cell double in size. S, primary activity is DNA synthesis and key event is DNA replication. G2 is the preparation for mitosis and the key event is protein synthesis. M phase is the division phase and key event is the physical separation of cells. Differentiation between mitosis and meiosis. Mitosis is occur in somatic cells. Meiosis occur in germ cells. Mitosis is the nucleus divides only once. Meiosis, in it, nucleus divides twice. Mitosis is divided into prophase, metaphase, anaphase, telophase, and the meiosis is the divided into prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, anaphase 2, and telophase 1, telophase 2. Daughter cells are diploid in mitosis, daughter cells are haploid in meiosis. No crossing over take place in mitosis, crossing over take place in meiosis. Increased number of normal cells in mitosis, produce egg and sperm cells in meiosis. Involved in healing and growth in mitosis, involved in genetic variation in meiosis. 2 daughter cells are produced in mitosis, 4 daughter cells are produced in meiosis. Malfunction result in cancer in mitosis and the malfunction result in genetic disorder in meiosis, such as Down syndrome, Knefter's syndrome, Turner syndrome. Quiescent stage G naught phase. Feature: quiescent stage, G naught stage, active cell cycle. In G naught, primary goal is specific cellular function, and the active cell cycle is preparation and execution of division. In G naught, DNA replication is DNA remains stable and DNA is synthesized as phase. In G naught, metabolic activity is high and the active cell cycle is also high. Duration in G naught phase is temporary and active cell cycle is predictable timed sequence. Some cells in the adult animal do not appear to exhibit division, for example heart cells, and many other cells divide only occasionally as need to replace cells that have been lost because of a injury or cell death. These cells that do not divide further exit G1 phase to enter in active phase called quiescent phase G0 of the cell cycle. M phase. M phase is the most dramatic period of the cell cycle, including a major reorganization of virtually all components of the cell. Since the number of chromosomes is in the parents and the progeny cell is the same, it is called as equational division. Here both chromosome and progeny number are same. So this is called equational division. Thought of convince mitosis has been divided into 4 stages of nuclear division. Karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut line cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase, telophase. Prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. Here, in prophase, initiation of condensation of chromosomal material, and the chromosomal material become untangled during the process of chromatin condensation. Okay, the centrosome, which had undergo duplication, during S phase of interphase, now begin to move toward outside opposite pole of the cell. The completion of prophase can thus be marked by the following characteristic event: chromosomal material condensation to form compact mitotic chromosome. Centrosome, which had undergo duplication during interphase, begin to move toward opposite pole of the cell. ठीक है. Metaphase. The complete disintegration of the nuclear envelope marked the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This is very clear that we can observe in microscope. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatid which are held together by the centromere. Small disc-shaped structure at the surface of a centromere are called kinetochores. Kinetochores is the disc-like shaped structure present in the surface of centromere. Uh, this is the phase where kinetochore is appear in the metaphase. Okay, these structures serve as the site of attachment of spindle fibers formed by the spindle fiber to the chromosome that are moved into position at the centromere of cells. Hence, the metaphase is characterized by all the chromosome coming to lie at the equator. In metaphase, all the chromosomes lie at the equator. Which one chromatide of each chromosome connected by its kinetochore to spindle fiber from one pole, and its sister chromatide connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as the metaphase plate. Okay, then metaphase having two features. First is spindle fiber attached to kinetochore of chromosome, and the chromosome are moved to spindle equator and get lined along metaphase plate through spindle fiber to both poles. It's very clear that in metaphase, first we observe kinetochore, and then second we observe movement of spindle equator in the middle of the cell anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split significantly, and the two daughters chromatid, now referred to as the daughter chromosome of the future daughter nuclei, begin their migration toward the two opposite poles. Here we observe that in anaphase, two chromosomes are moved toward the poles, future daughter nuclei begin their migration toward the two opposite poles as each chromosome moves away from the equator plate. This is very common. After metaphase, anaphase, where we observe movement of chromosome opposite direction of the equator toward the pole. As each chromosome move away from the equatorial plate, the centromere of a cell chromosome remain directed toward the pole and hands at the leading edge, with the arms of the chromosome traveling behind. Thus, anaphase stage is characterized by the following key events. First is centromere split and chromatid separate. Second is chromatid move to opposite poles. After anaphase, we going toward telophase. At the beginning of the final stage of karyokinesis, example telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen, and each set of chromatin material tend to collect at each of the two poles. This is the stage which shows the following key events. First is chromosomes cluster at opposite spindle poles, and their identity is lost as the discrete elements. Second is nuclear envelope develops around the chromosome cluster at each pole following two daughter nuclei. Nucleolus, Golgi complex, and here is reform. After anaphase, we are discussing telophase, in which chromosome clusters go at opposite spindle poles, and formation of nucleus Golgi complex in ear reform, and the nucleus envelope develops around the chromosome cluster at each pole following two daughter nuclei. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei, but the cell itself is dividing into two daughter cells by separation of cytoplasm called cytokinesis. Okay, cytokinesis is basically separation of two daughter cell in cytoplasm. In an animal cell, this is achieved by the appearance of furor in the plasma membrane. In plant cell, all formation start in the center of the cell, grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represent the middle lamella between the wall of two adjacent cells. Significance of mitosis. Mitosis, or the equational division, is usually restricted to the diploid cells only. However, in some lower plants and in some social insect, haploid cell also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insects? Mitosis usually results in the production of a diploid daughter cells with identical genetic complement. The growth of multicellular organism is due to mitosis. So growth of multicellular organism is the main response of mitosis. Cell growth result in distributing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio between the nucleus and the cytoplasm. The cell of the upper layer of the epidermis, cell of the lining of the gut, and the blood cell are being constantly replaced. Mitotic division in the meristoplasmatic tissue, the apical and the lateral cambium result in the combination growth of plants throughout their life. In meiosis, we study meiosis 1, meiosis 2 parts. Meiosis involves two sequences, cycle of nucleus and the cell division called meiosis 1 and meiosis 2, but only a single cell cycle of DNA replication. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatid at the S phase. Meiosis involves pairing of a homologous chromosome and recombination between non-sister chromatid of a homologous chromosome. 4 haploid cells are formed at the end of meiosis 2. Mitotic event can be ground under the following phases, as I before said that meiosis 1 and meiosis 2. In meiosis 1 we study prophase 1, prophase 2 and meiosis 1 have contained the prophase 1, metaphase 1, anaphase 1, telophase 1, and in meiosis 2 it contains prophase 2, metaphase 2, anaphase 2, telophase 2. First we go toward meiosis 1. Meiosis 1 contains prophase 1. Prophase 1 is the prophase of the first meiotic division, is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior. First is a leptotene, the zygotene paracrine, diplotin, and dikinesis. During leptotin stage, the chromosome become gradually visible under the lightest microscope. It is very clear that leptotin is visible under the light microscope. The compaction of a chromosome and continued throughout leptotin. This is followed by a second stage of prophase 1 called zygote. Zygote is the stage of chromosomes start pairing together. Zygote is the stage where pairing of chromosomes take place. This process of association is called synapsis. Synapsis is the formation in take place in zygote. Such paired chromosomes are called homologous chromosome. Electron micrograph of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex. Also, it's a very important point where is this complex form. This is the major point where zygote is the place where synaptonemal complex is formed. This complex is formed by a pair of synapsed homologous chromosomes is called a bivalent or a tetrad. Also, that zygote is the place where tetrad is formed. However, these are more clearly visible at the next stage. The first two stages of a prophase 1 are relatively short-lived compared to the next stage, that is paracrine. During this stage, the four chromatin of each of bivalent chromosomes become distinct and clearly appear as a tetrad. This stage is characterized by the appearance of recombination nodules. The sites at which crossing over occur between non-sister chromatide of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called recombinase. Recombinase is a place where it occur in paracrine. Crossing over lead to recombination of a genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of paracrine, leaving the chromosomes linked at the site of crossing over. The beginning of a diplotin is recognized by the dilution of the synaptonemal complex and the tendency of the recombinant homologous chromosome of the bivalent to separate from each other except at the site of crossover. This X-shaped structure is called chimata. In oocyte of some vertebrates, diplotin can last for months or years. The final stage of meiotic phase 1 is dikinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosome for separation. Metaphase 1, the bivalent chromosome align on the equator plate. The microtubules from the opposite poles of the spindle attack to the kinetochore of homologous chromosome. Anaphase 1, the homologous chromosome separate while sister chromatid remain associated at the same centromere. Telophase 1, the nuclear membrane and nucleolus reappear, cytokinesis follow, and this is called as diad of cells. In metaphase, we observe that separation of spindle fiber form microtubules from the opposite pole. of the spindle, and in anaphase we observe that separation of two sister chromatid and associated at their centromere. In telophase, one nuclear membrane and nucleus reappear, and cytokinesis follows and is called a dyad of cells. Interkinesis is followed by prophase 2, much simpler prophase than prophase 1. Meiosis 2 contain prophase 2, metaphase 2, anaphase 2 and telophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes may have fully elongated. In contrast to meiosis I, meiosis 2 resembles normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact. Metaphase 2, at this stage the chromosome align at the equator and the microtubules from opposite pole of the spindle get attached to the kinetochore. And phase 2, it begins with the simultaneous splitting of the centromere of each chromosome, allowing them to move toward opposite pole of the cell, shortening of microtubules attached to kinetochores. Telophase 2 meiosis end with telophase 2, in which the two growth of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells. That is form 4 haploid daughter cell. Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexual reproducing organisms. Even though the process per se paradoxically results in reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variation are very important for the formation of evolution.